And next up is going to be Dr. Thi Nguyen from Pittsburgh, who's going to share some of her experience with repositories and challenges and opportunities.
Okay, good afternoon. Thank you for inviting me to give the talk today. So I was asked to discuss about the opportunities and challenges in establishing the invasive fungal infection biorepository. To structure my talk, I'm going to use my experience with 4 biorepositories that I have been using to guide me with my translation and research. For the lack of time, I'm not going to discuss much about the multi-center candidemia repository, except to tell you that this repository have about 500 candida bloodstream isolate that are linked to epidemiology data as well as the treatment data. I will spend most of my time to talk about the NIH-NIAID funded Mycology research unit to myself and Neil Clancy when we were at the University of Florida. And the title for this NIH MRU is In Vivo Induced Antigen Technology. So basically what we do is that we take the patient's human sera from patients with candidemia or any kind of candidiasis, like esophageal candidiasis, and we use that serum to hybridize the candida library. And what we are looking for are immunogenic antigen that are expressed mainly during the infection and not in vitro. Our hypothesis is that these antigen will be a very good, serve as a very good markers for virulence, pathogenesis, as well as biomarkers for diagnostic and prognostic study. So this MRU has a full independent project. We have our own share goal is that we use the biorepository that are established by John Wingard when I was at the University of Florida. So we were very productive with this MRU with multiple publications, including the one in molecular, micro, as well as blast pathogen. So in the middle of the IRU, the NIAID announced that they no longer favor the routine funding of the program projects, including all the MRUs that are currently funded. So what I did was I used my own funding to continue with the repository at the University of Pittsburgh. I'm going to share with you the most recent study coming out from this MRU. So the basic question is that we know that the burden of entry for candidemia is mainly from the GI tract. GI tract harbor like a 10 to the 8, 10 to the 12 CFU and candida. So once the candida get into the bloodstream, does it make sense? My question is that the blood culture with candidemia, is it due to a single organism or due to a population of candida? And in order to answer that question, we took the blood culture from patients with candidemia, we shrink it and randomly pick 10 strands for whole genome sequence. We did that for both Candida albicans and Candida glabrata. Candida glabrata genome is a little bit complicated because they have a lot of gene duplications, so we ended up using both long read and short read sequencing. So the figure on the left is the phylogenetic tree of what the 100 Candida glabrata isolate from 10 different patients, and from each patient we use 10 strands that we randomly pick. The figure on the right is the heat map looking at the distance, and you know when the color is blue, dark blue, it tells you that the genome are very well related. But when it's become lighter, yellow or orange, these are very far related strands. 2 things that you can take home from this slide. Number 1 is that each patient with candidemia were infected with their own strands. But what most interests us is that when you look at the individual strain within the same patients, you can see here all of these are genetically distinct. So, to make your job easier, I present in the circular phylogenetic tree and also on the Venn diagram of the individual strain from the same patients. And you can see here, there is like a distance between individual strain from the same patients. And what we also found was that we did a broad phenotype study, including the antifungal susceptibility for all of these 100 strains. And what we found was in 2 patients, the patients were actually infected with two different strains of Candida glabrata that differ in the azone susceptibility. And none of the azone-resistant Candida glabrata was detected by the microbiology lab. One patient especially was actually reinfected after 5 weeks of treatment with caspofungin followed by fluconazole. And the strain that caused the reinfection actually was present in the original blood culture that was missed by the clinical microlab. So to understand the mechanism of azone resistance, we treat these organisms and then we do follow-up study. On day 3 of the incubation, we start to see the development of small colony variant. It suggests to us that these might be due to mitochondrial defect. So what we look at is these are the cells, individual cells with mitochondria in it. Unlike the normal colony variant, the small colony variant has diminished in the number of mitochondria, and the mitochondria structure looks very abnormal. Candida glabrata has 13 chromosomes, one of which is mitochondria. So instead of looking at the entire genome, we zone into the mitochondrial phylogenetic tree. As you can see from the tree, at least the 5 small colony variant, there are numerous mutations within the mitochondrial genes. So on the second patient with the mixture of azone resistant and susceptible, so these isolate, we call it L1 to L10. 2 of these isolates were resistant to azone and cross-resistant to other non-active azone as well. Looking at the whole genome sequence, we found that there is a mutation within the PDR gene. PDR gene is a transcriptional regulator of the CDR1, which is the ABC cassette transporter gene. And this is the mechanism of resistance, azone resistance for this patient trend. So based on this study, we think that in some patients with candidemia, unrecognized fluconazole-resistant strains are present in the minority population of blood because the clinical lab only pick a representative single colony, then these minority azone-resistant might be missed. Our findings actually challenge the long-standing paradigm that most bloodstream infection was caused by a single strain. Our data suggests, on the other hand, that we are dealing with a population of microbiome genetic diversity during the bloodstream infection. And the clinical relevance of this observation will need to be established in the future study. Now, I would like to switch the topic to discuss about the second biorepository that I'm working with John Wingard as a PI for the NIAID-funded U01 Aztec. There were 2 objectives of this study. Number 1 is to establish and maintain the repository with the patient sample at high risk or diagnosed with invasive aspergillosis. And the second aim is to establish a network of 5 laboratories compliant with the good laboratory practice to assess the new diagnostic assay for invasive aspergillosis. And the 5 labs are represented in this figure, and Borber Alexander oversee the entire process. So for sample collection, there are 2 complementary strategies. Strategy number 1 is to look at, enroll longitudinal collection of sample for patients at high risk for IA. As you can see, this approach is very inefficient because the high-risk patient, only about 10% of those would end up developing IA. The second strategy is to collect samples from patients with proven or probable IA, and the limitation is that there is no baseline uninfected sample from these patients. So Aztec, was very aggressive in obtaining the sample. You can see here they obtained from whole blood, leukocyte, serum, plasma, and also RNA. In addition, they also collected BIENNIAL sample, urine sample, and other samples that are presented on this slide. So through the 5 years of funding, 872 patients were consented into this study, and 187 of them developed proven and probable IA. There were a lot of accomplishments from the Aztec from this U01, and all the accomplishments are summarized here. Basically, I would like to point out that the Aztec created the largest invasive aspergillus biorepository that I know of, and this is provide a unique resource that no one else available. And there was numerous publications out of this study. So lastly, I would like to briefly discuss about the biorepository number 3 that I just started. And the title of this is Don't Forget the Environment. We know that the environment can serve as the source of fungal infection to the high-risk patient. One of the examples is that the agriculture origin of ozone-resistant aspergillus, and patients who are infected with these have a very bad prognosis. And the second example is the evaluation of our outbreak of mucormycosis back in 2015. We found out that the environment, especially the linen, might serve as a source of it for this outbreak. And we collected at least 100 isolates during this investigation, and they all underwent whole genome sequence. And we showed that there was a genetic diversity in both the clinical as well as the environment sample. So in December of last year, the CDC made an announcement that they accept the applications for applied research to address emerging public health priorities. We submitted our proposal entitled Surveillance of Pathogenic Fungi in Hospital Environment and Patients. And I was very happy when we heard that this project was selected by the CDC for funding, and we cannot wait to get the study started. So lastly, I would like to discuss the challenges with the fungal repository. The first thing that came to mind is the cost. The reason that we were able to do the MRU repository and Aztec repository is because of the very generous funding from the NIAID. Costs will include paying staff, equipment, software and hardware maintenance, data management and sharing, service contract, as well as supplies. And the job doesn't stop there, because we still have to come up with funding for the long-term commitment in maintaining this repository. So several centers in the US make up some kind of financial management system, and most places actually implement a cost recovery system. Actually, they charge the investigators and company to access these specimens associated with data, and despite this adoption, the majority of the repository are unable to fully recover the cost. And there are major ethical and legal issues with running the biorepository as a business unit. Other limitations, including giving the reconnection to the PI who actually established this repository, because it takes a lot of time and effort. And people started to talk about shared authorship, although this has been underdeveloped. And another challenge is that the life cycle of this sample is not known. We know that the sample may be good for up to a year, but whether they stay good after a year or not is not known, and we have to constantly retesting these biorepository. So, in conclusion, the repository is an integral part of medical research. Infectious disease and mycology has lacked other disciplines such as oncology. The success of the repository will rely on patient participation, the number and diversity of available biospecimen, cost management, and most importantly, national and private sector funding. Thank you very much for your attention.